Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy. Today on the Microscopists.
Hi, this week on the Microscopists, I meet Anne Carpenter from the Broad Institute of MIT and Harvard as we discuss surviving imposter syndrome.
Does it matter if I'm the best scientist in the room or the smartest person or what? Like, does any of that really fundamentally matter, or are there other aspects of my life that give me meaning and value?
Overworking microscopes.
They sent another one, and then within a week it was burnt out again. I called them back and they're like, "What are you doing with that shutter?"
The simple joy of chocolate chip cookies.
I've made, it's gotta be in the thousands of batches of cookies at this point in time.
And the best way to get round nose to tail eating when growing up on a farm.
Can't remember. I think we always gave all the weird bits to my grandmother. So I don't recall ever having any of the sort of funkier parts that a person might want to eat.
All in this episode of the Microscopists. Hi, welcome to today's episode of the Microscopists, and today I'm joined by Anne Carpenter from the Broad Institute over in the US. Anne, good morning, good evening, good afternoon, whichever time of day it is. How are you?
I'm doing well, thanks.
So, Anne, firstly, thank you very much. I guess one of the things you're most famous for is Selfprofiler, and as a microscopist running a core lab, lots of users, we've pointed lots of users in the direction of Selfprofiler as the platform to go to for large cell analysis. Even my PhD student was actually recently on a course which was co-run by yourself at EMBL. Yeah, so yeah, it's been a huge asset to us. But I guess to many listeners, they may not even, or viewers, they might not even understand the importance of image analysis, which seems really easy, doesn't it? You just look at a cell and you analyze it, but it's not. So Anne, give me 2 minutes on this, because actually I think so many people do not understand the difficulties in image analysis. And we'll come to lots of other things later, but it'd be great to hear why this is so important.
Well, I think it's something that the younger generations of scientists are kind of born in a world where you quantify images. Some of us grew up in a time where images from microscopes were just either observed by eye or just one snapshot is what makes it into the paper. But I think it's increasingly becoming the norm that if you see something by eye, that's not enough. You really want to quantify any behaviors that you're seeing in samples. And so it's important, of course, for reproducibility and for being sure that you're not fooling yourself of what you're seeing by eye. But then the domain of science that my laboratory tends to focus on is this high throughput microscopy world, where it's just an absolute necessity. That it's when I was first starting my career, people really did look through piles and piles of images, often hundreds of thousands, in order to do a screen. People certainly did that by dissecting fly eyes and looking at fly wings as well. But these days, it's really becoming quite standard to quantify, and especially in the high throughput world, there's really no serious alternative these days to quantifying images.
Which is obviously really important for research, but also for pharmaceutical companies, for drug trials, and just seeing global analysis. We think of when you add a drug or a compound to something, we need to see the changes. And actually, quite often we can see that by our naked eye, or some changes within that population, but quantifying it, well, surely if I can see it, it's really easy. No?
That is true. If you could see it, the quantification should be pretty straightforward. And I think we really can say that now. I think 10 years ago, it was still the case that a lot of phenotypes were challenging, a lot of organisms were challenging, a lot of cell types were challenging. And now out of the pie of all the things you might want to measure, I would say it's really the proportion that is difficult is really pretty, it's becoming a small sliver at this point. So if somebody's working with kind of a standard mammalian cell type, most phenotypes, if you can see them by eye, they can be not just can they be quantified like by collaborating with a computer scientist for 6 months, but they can be quantified using off-the-shelf tools that are really convenient and easy for any biologist to be able to learn how to use.
So I've got, I'll come back to you after this meeting because I've got loads of label-free quantitative phase images that we are still struggling to really get.
Okay,
everything.
so that's part of the sliver that's still a little challenging. So I will say, certain cell types like neurons can the signal to noise and it's quite a challenge, and bright field phase imaging can also be a bit of a challenge, but we're right on the cusp, especially with deep learning methods with bright field and phase images. We're just right on the cusp of these becoming more and more tractable.
Yeah, I think the quantitative phase images are pseudo fluorescence in a way, but anyway, we will talk about that later. But that comes to an interesting point. You introduce yourself and you're talking about computer scientists, but that's not where you started out life. And I think that's really valuable to us as biologists, that you are one of us. You're a life scientist. So I think you had your biological sciences degree at Purdue.
That's right.
And then a PhD. So I did some background research over at Illinois Champaign. Is that correct as well?
Yeah. You got my credentials. Yeah.
But okay, so that's fine. That's into molecular biology. How did you get into computer science?
I think out of necessity is the short answer to that question. So for my own project that I needed to accomplish during my PhD, it became clear that I was going to need to, it's just too tedious. It was literally holding up a ruler to the screen of the computer to measure things. And I said, oh gosh, there's got to be a better way, because I don't want to fool myself to thinking I'm seeing things that I'm not, and so on. And I really wish this could be a bit more objective and automated. And so I started dorking around a little bit with Image-J at the time, NIH Image at the time, and just really fell in love with it just feels like magic, honestly, when you take all these messy images and each one is a little unique and has some funny things going on with it. But then when you take dozens of them and you extract all their metrics and you end up with nice little scatter plots of data, to me, it's really a beautiful thing to be able to turn the kind of messiness of biology into something a little more measurable.
So from my side, I used to measure things with rulers, and I'd still be tempted to measure things by rulers, but I guess that's very old school now for some bits. How do you make that transition though? Because that's not an easy switch, because then you built a tool or tried to develop a tool to solve that, but then that became your career. So you kind of moved out of the life science and into this huge asset and the computer science side. How do you skill yourself?
Yeah, so I think the reason I was successful in that transition is because I didn't realize I was doing it at the time. If I had known that was ahead of me, I'm not sure I would have been so brave as to make the leap. But I had that exposure during my end of my PhD for my project, and then during my postdoc at the Whitehead Institute with David Sabatini, he was creating these live cell microarrays that were producing image data at a scale that was previously not common. And so it was clear in that project I was going to need some automated analysis. So I really just started by surveying the existing tools and found nothing was going to do what I needed. And so I found a couple of papers that I could scarcely understand, but were from the computer science literature that looked like they had algorithms that would help me. But as you might be aware, computer science papers almost never have code that go with them. Even if they did have code, it's unclear whether I would have been able to execute such code on my own at that time. So I just reached out, literally a mass email to the MIT computer science department grad student list, and just said, I need help on my project. I have some fellowship money. I can pay you hourly for your time. Can anybody help me implement this algorithm for my project? And connected with Thoas, Ray Jones, and he implemented the algorithm in a weekend. It was very straightforward for him to be able to do it. And that really started, seeing the effect of that on my project really got me hooked. And so we ended up collaborating. He ended up switching his thesis project to work entirely on this. And over time, I ended up focusing more and more on the software and less on any particular biological problem. I started helping everybody in the departments around us working on their projects, and the software tool, which I never intended to become a major thing. It was really just for me and my friends, so to speak. Eventually, it became clear that this was filling a niche for automated high throughput analysis that wasn't being served by the existing software.
So, and that software, I presume, is CellProfiler.
Yep.
At this point. So you said you built that for you to use, and then obviously your friends started using it because they saw the benefits of it. Now it's used by almost everywhere. Most universities across the world will be using this. The impact is huge. It's free to use. It costs nothing. Do you regret not commercializing it?
Not one bit, not one bit. And I mean, for two reasons, one noble and one practical. The practical one is that it's not easy making a living trying to wrench dollars out of biologists, right? I mean, given a choice, most biologists are like, oh, cost $15 for this thing, or I could just, sort of finagle it myself in some other way. So I think partly it's really just the practical, that it's hard to sell software to scientists in general. We're sort of used to everything being kind of free or coming with the microscope, or it's not easy. And I think if you buy commercial software, hearing people complain about the cost of commercial software, I mean, it's just the reality. That's how much it costs to have the marketing department and the actual engineering that goes into a tool and so on. It's an expensive enterprise. We're a small community, so things are going to cost quite a lot. So I didn't think it was very practical at the time. But then, of course, there's also the noble reason, which is it just seemed to me at the time, the world doesn't need another commercial package. The world needs, for this field, which at the time was just about to take off, this whole high content imaging, really high throughput imaging field, it just seems like this is what the world needs right now, is something open that can be readily adopted. And I think if I make it open, it'll be very well taken care of. So I think the things that have made me slightly regret the choice is when I try to obtain funding for the software. So every day a couple of papers come out that cite using the software. It's very useful across the community, and it's not a lot of money. Usually just one software engineer to maintain a software project of this scope. It's like an incredible bargain from the funding agency's perspective, but at times when grants have come through rejected, I've been like, if only we could get some amount of just a little bit of income to make this thing support itself. That would be maybe that would be a way to go, or maybe we'll splash ads on the software from time to time. It's been a little bit of a struggle to try to figure out how to keep it funded, but I do have to say we've been extraordinarily. We're just really grateful for the support of mostly two agencies over time. One is the National Institutes of Health, which is the major US funding agency. And then also surprisingly came along, the Chan Zuckerberg Institute has adopted this bioimaging ecosystem as one that they want to invest in. And it really couldn't have come at a better time, because it's a real struggle otherwise to make software engineering, financially solvent in the academic world.
So I think that's interesting to hear, because you're hugely successful. You're at the Broad Institute, you're Harvard, MIT behind you, but it's still not a given. You still have to fight to get even the basics kept running. And the impact, I think actually, if you'd have commercialized it, you would not have had the impacts across science that you've managed by having it freeware. I think that actually that route, I think there's a world for both commercial and freeware. Neither is wrong. And I don't see the commercial world as evil. Far from it. There can be lots of support. As you say, it's keeping it running and supporting. That's why there's a cost to it. But I think your impact across science, and it's not just biology or microbiology, it's a broad impact that you've been able to have because you went to freeware in this instance, which is terrific. But it's great that you've got funding now, because I was going to say, it's amazing that some of the big pharmaceuticals don't club together and just holistically put some money in, because they use it. They're big benefactors from it.
That's right. And well, our software project is quite secure at the moment. There are other software projects that aren't, and it's amazing to me that we haven't figured out a decent mechanism to make that work, right? Because again, a software engineer to work on a project is typically all you really need. You don't need 20 or 30 of them working together on most of these projects that are so useful in the scientific community. So even just funding one of them, it's 10 or 15K from a dozen companies that make use of the software would make most of these software projects be very, very happy. But we just haven't quite figured out how to make that work in more of a social, cultural kind of way. Like pharma company, send me an invoice, I'll pay it, but I don't really know how to sort of make a donation to something that I value.
So it sounds from that, so we've got to be careful here that it's you and one other person, and it's far from that. And I can't find that picture of the background for your team. So how big is your team?
Yeah, so typically my team is around 10 people, but only less than half of them in the past few years or so, less than half have been focused on image analysis per se. My group is moving a bit more towards the data science and machine learning of image-based data. And so that I would call is very separate. So as far as the actual software engineers for the Selfprofiler project, it's almost always been around, on average, it's been one software engineer for the past 14 or 15 years. I was the first quasi software engineer, and Ray Jones as well. But that's not a huge undertaking. But you're absolutely right. It's not a one-person effort in the real sense, because we also have in my group alone, we have 3 or 4 people who use the software all the time. They're engineering savvy enough to identify problems and in some cases fix individual bugs that they encounter in their own work. And so there's the saying in the software world that we eat our own dog food, that we have to deal with the software as it is, and of course that makes us want to make it constantly better. And so yeah, there's about half the group that's focused on image analysis. Most of them are focused on applications and not necessarily the engineering of the software itself. But it's a very blurry line because a lot of folks in the group work across both sides.
So just thinking about your team, I've met Beth a few times. How is Beth?
She's doing very well, thank you.
Yeah.
Cool. So that's Dr. This is Dr. Beth Simmons. She's been with me ever since around 2016 or so, and she's now leading this side of the group that I just described, the whole image analysis. She launched and runs a bioimage analysis training program that is our mission. Well, I'm sure there's an official mission statement somewhere, but the way I see it is it's a method of turning biologists and microscopists into employable image analysts. So moving people more towards the computational direction where they can really have a meaningful career helping people do image analysis.
So it's a big team, and so they're your children, but actually I think this picture here. So
there we go.
Can you describe who is in this photo, please?
Yeah, so those are my 5 kids. I have the 3 older are my stepkids, and the 2 little ones are my biological kids. And yeah, I was just thinking about it this morning that I acquired them over a 5-year period. So went from 0 to 5 in a pretty short period of time, kind of high throughput there. And it's definitely been just an incredible transition in my life, I guess it's the way to say it.
So you haven't segmented them yet and started tracking each individual. It's still in that 5-year high throughput.
Yeah.
Analogy. So how old are they all?
The youngest is just about to turn 7. Yeah. So, and then the oldest is an employed teacher, just finished college out in Oklahoma. Yeah.
That's cool. And how? So. You had that 5-year spurt, so we're talking youngest is 7, 5 years. Oh gosh, that's 12 years ago. So you're going to have been quite early in your career still.
That's right. So yeah, all of my children were acquired, let's say, just after I started my position at the Broad. And it is a funny question people ask, like, when is a good time to have children? And I think I think you should probably not think about your career when deciding this question, because there's just pros and cons for every career stage. I think the advantage of having kids a little on the later side, when I had my independent PI position, the advantage there is I had enough money that I could hire housekeepers and could not worry so much about every little expense and because a lot of times you just spend a lot of energy and time when you could replace that just with a little bit of extra money. And so for me, it was wonderful to have kids during that period of time. On the other hand, you could argue, well, the first first couple of years establishing my lab is just incredibly intense, and it's sort of it's a pretty anxiety provoking time. I mean, of course you're anxious during the PhD. Am I going to get a good position afterwards? And during your postdoc, am I going to get a good position afterwards? There's anxiety throughout the spectrum. But I think when you're starting a lab, like, this could sink or swim. You really don't know if it's going to work out well, and you don't know to what extent is your vision for a lab really going to play out? Are the grants going to get funded? Is this really going to work out? So that was not the ideal time to be up all night with babies and so on, but it worked out. So likewise, there's all kinds of advantages to having kids in a PhD or postdoc period, because your schedule's just, you're not responsible. If your project pauses for 6 months, no one in the universe is going to have a problem with that, right? It affects your productivity overall for sure, but you've got a 3 or 4 or 5 year time span depending on what stage you're in, and but on the other hand, you have no money, so it can be a challenge that way as well.
Yeah, I was going to say, I think there's a lot of PhD students and early postdocs that would love to have children, but financially can't take that risk at that moment in time, and they know it's quite a nomadic life. For quite,
exactly. You don't have
academia, people move around cities, countries, continents.
Yep. So not only do you not necessarily have your nuclear family of origin support or extended family around, but you may not even have friends around because you've just moved to one location or another. So that's a real challenge of the typical scientific life. I mean, of course, you can make sacrifices and decide to stay in a certain geographical area, but it's a sacrifice. It has some trade-offs in some other directions as well.
So how did you balance having the children? How did you even segment your thought process to work is very we are very focused people. How did you partition that, or how did you merge them?
Yeah. So for me, it was actually it was not a terribly difficult time, and I think this advice is very applicable to just about anyone. So well before I had children, back to the very first days of my PhD, I had decided that I was going to work during work hours and I was going to not work during not work hours. And those work hours were typically 45-ish, sometimes 50 or 55, but never 80 or never, well, okay, occasionally they would just spill outside. But by kind of enforcing that really early on in my career, when it didn't matter, I didn't have anything to do outside of work hours, but volunteering and entertaining myself. But I think because I took that kind of structure really early on, then when I had children, suddenly I had no leisure time outside of work. It went from all work and all leisure to all work and all kids. But at least it wasn't trying to shove a 70-hour work week into 40 hours or something like that. So for me, I would say the transition was not as difficult as it would have been if I had let work spill over in the first place. And so I'm not going to say that it's trivial. There's just a lot of logistics to a family with that many children, and some of them have some special needs that are a little bit challenging at times as well. And so it's definitely not trivial to I am pretty much doing something for somebody at every waking moment of my life, but a little less so now that my youngest is 7. I actually do have leisure time again in my life, which is pretty exciting. So it's not easy, but I think it's a lot more manageable if you have kind of a bit of put limits on your work life before you have children. I think it's a better transition.
So you mentioned leisure time. What do you do in that leisure time?
Yeah, so I'm not that interesting. There was a TV show. The public television station wanted to do a TV show about women in science, like more than a decade ago now, and they sort of asked me this question, and I went through my list of hobbies. They're like, "You got anything else? You got anything else?" Like nothing interesting to televise, I guess is the answer to your question. So my hobbies are reading, baking, sitting around chatting with my friends, dinner parties, like this kind of thing. So yeah, nothing too dramatic or interesting. I mean, I guess I would say now my leisure time is spending time with my kids and doing fun things, going on hikes and stuff like that. There's the baking. Yep.
Yeah. So there's one of your youngest, is it?
Yep.
Yeah, so actually, so you are now doing baking and looking after your children at the same time.
Yep. Multitasking is the name of the game. That's right.
I think we all have a photo of our children baking at some point, just to remind them that when they were young,
so
they wanted to, because they don't anymore. Actually, one's come back from university and decided that maybe his cooking's better than ours. So maybe when they leave, they decide that actually our home cooking is no longer the best that's out there. And this
interesting.
Here,
so this is a Valentine's Day aftermath. One of the things that's been really fun with my kids lately is they've gotten. I mean, I don't know how to convey this. I don't want to make it sound like, oh, my children are scientific geniuses and every scientist's kids should also be, because this is actually a fairly new development. But it was hilarious. We got some kind of science-related handbook and a little lab notebook for Christmas. And so when we got some flowers for Valentine's Day, the first thing the girls wanted to do was chop them up into bits and name all the different parts. So this is the aftermath of Valentine's Day at our house just a few weeks back.
That's pretty cool. So have you put slices of these or elements under the microscope so they can see it in more detail yet?
Oh, that's a great idea, but no, we haven't. We do have a little teeny toy microscope laying around. Yeah.
You just need to find an old one that works throwing away and bring it home. I haven't I haven't done that. I hated microscopy as a child, so they will find it in their own time.
Yeah, and I'm definitely hesitant. I hate the idea of kids feeling like they're pushed in a certain direction, and so we've tried to stay pretty open-minded. I mean, I think I'm adamant about my kids being curious and like, you know, kind of questioning things about the world and pursuing things that interest them for sure. But I think I actually mostly hesitate away from science when they're expressing their interest, just so that they don't feel overly shoved in that direction.
Yeah, no, I think that's a wise decision. And actually, when they get smarter than you, it's quite difficult. That's a learning process. When some of them, really smart cookies, and it's just like, well, I don't know.
Yeah. I'm bracing myself already. That's why I'm also teaching them to be sort of humble and kind to those that are maybe not as bright as they are, because that's probably going to be me someday.
Yeah, see, my eldest is actually a computer scientist. That's what he's studying at the moment, and maths with computer science, and that's why I've got so much admiration for yourself, because I just couldn't do it. Just completely lost on it, and it's. Yeah, he does it. So actually, when I've got problems now at work, I can go to him.
Excellent.
And he's actually quite keen, but he hasn't seen the light when it comes to putting his skills towards biology yet.
Yeah.
So he hasn't quite seen, because everything seems really trivial. He's going to watch this, listen to this, isn't it, at some point? Be like, well, that'd be easy, and I know it's not that easy from the tasks I want to be done, but. Anyway,
and that is a real challenge for getting computer scientists into this field, is that it's very application oriented, and that just the reward system built into computer science departments does not reward applications. It rewards interesting theory and sort of clever mathematics. And so that is a real challenge, is getting folks to be interested in working on problems that are sort of too easy for them. It's a real problem.
Yeah. But they're not that easy. Not when they get down to it. They're just conceptually easy until you want to do something. It's like, oh,
in practice, in practice, it's easy enough to get something to work on one image or a handful of images, but then when you've got thousands, and I think actually that's why Selfprofiler has been really popular and really successful, is first of all, it was designed by a biologist who needed it, so it wasn't designed by somebody who had no clue what our typical workflow is like, and secondly, it was designed for the worst case scenario, which is you've got to run on 100,000 images and not tweak each one to make it work, right? It was really designed to be robust to a large image set, which then I think also translates to being robust across different laboratories, experiments, and so on.
So I will come back to that in a minute, but you said a minute ago that one of your pastimes, for leisure, you enjoy reading. So what sort of reading? What are you into?
Oh, sadly, Oh, that's embarrassing because since I've had little ones, I would say almost all of my reading is on my phone, and it's either Twitter or articles linked from Twitter. It has to be the vast majority of my reading material these days, just because it's just bits and pieces of time scattered throughout the day. Before that, I don't know. Really the spectrum. I like historical fiction, historical nonfiction. Novels is really quite a broad, just theology, like all kinds of things is what I used to read in the old days when I had, the mental bandwidth to focus on something for a few hours on a Saturday.
It's interesting. I have noticed you are a prolific retweeter on Twitter itself, which is, and you support a lot of initiatives. So you do promote a lot of positive messages on there as well. So yeah, yes, I'm a follower. So I
was just thinking, my bio in 2016 or maybe 2015, I put, I will stop tweeting politics soon, because I felt so sheepish of that aspect. But over time, with the politics in America being what they are, I finally just took that out, because it's probably not likely I'll stop tweeting politics. But I'm glad you're focusing on the other stuff, which is the kind of trying to. I think it's really heartening to me to see a lot of PIs, I'm not going to say my age, I just mean kind of a new cohort of PIs, whether they're older than me or younger, really making an effort to make science, I'm not going to say a nurturing place, but at least a place that is less toxic. Like, can we at least start there? And hopefully more so making it a place where people can do their best work. I think there's a lot of, I don't think science selects for weirdos, but it certainly doesn't weed them out along the way in the same way that a lot of other careers do. And I think there's a lot of behaviors that just wouldn't be tolerated in other environments. And so I really see this happening in the PIs that I talk to, that I interact most with, this real enthusiasm for creating, a better place, like the kind of place we would have liked to train, that's more drama-free than what a lot of people have been subjected to in their careers.
So and I think Twitter, for those who don't follow Twitter, actually in the academic world, certainly the life sciences, it's a really good way to keep on top of your subject. There's loads of good stuff out there that comes out in snap bites, easy to digest, and you can delve deeper because the links are there to delve deeper. I think different platforms are good for different purposes. I also use LinkedIn, but actually that tends to grab a different community compared to be
a bit more of the industry focused as well. Yeah, I don't see much on LinkedIn, but whenever I do poke in there, I'm impressed by the degree to which it captures that community really well.
No, and yeah, precisely. And it depends on what you're messaging or what you're wanting to read. And I think there's still a sort of a misunderstanding. Twitter, certainly celebrity-wise, is all about their conscious of thought. And I think in the science world, it's not about that mostly. It's about messages, information. You can almost see whole publications printed with the chains on it. It's like, oh my goodness.
That's right.
Ricardo Henriquez might have posted 27 in one or something once. And it's like, this is brilliant. I can see all the figures, all the information.
Yeah.
It was
so it's a scientific content, and then it's also just sort of critiques and movements to shove the community and the culture in a certain direction. So for example, just peer review and open science, open data, open publications. There's just a lot of peer pressure on Twitter for making science a more open place, which I think is a really great direction to be going. So it's definitely a segment of the society. It doesn't reflect really all of science, but it for the most part reflects a direction of science that I'm excited about, and I hope in general that we move more towards.
Yeah, so actually I think for the people listening, watching, get on there. I think there's a lot of resistance because they see it through different eyes. Follow the right people, put in the right tags, and you will learn loads. It will broaden your horizons, your literature. It will do your literature reviews for you, almost your searches for you to many respects. So thinking about publications and literature reviews, what's been your favorite publication that you've authored or co-authored?
I have to say probably the CellProfiler paper, which was the product of my postdoc, was one that I was the most proud of, because, you know, just every little being so intimately involved in every little figure and every little chunk of data that ends up, you know, each of it has a bit of a story behind it. And what was really nice about that paper is that I thought it was important to demonstrate that it was useful across a wide variety of science. So that meant as I said, all my friends who had different projects in different areas kind of came together and were in that one paper. For a more recent one, it's hard to pick a paper, but just the whole cell painting, series of papers, I think would be the one that I'm most proud of as well more recently. And there it's just because we're now, the original assay was developed, I guess it must be about a decade ago now, but now we're just starting to see the fruits of it being adopted in the pharma industry, and we're starting to see drugs going into clinical trials that were discovered using this approach, and it's really been tremendously satisfying for me.
So I think, again, we sort of touched on this at the start, and I think it's quite something that your impact now is on drugs and what could end up helping society in general. And that's never where you started this out. And this is the importance of blue sky research to an extent, and encouraging more niche research. I'm not saying what you're doing was niche, but, you know, focused. But the impacts, the benefit, we'd never envisaged this impact then. And the impact that it's had now is huge. And I think that's, people I don't think realize the importance of blue sky research.
Yeah, for sure. You know, one phenomenon I love to see is no matter what random crisis is going on in the world, whether it's COVID, whether it's you know, killer wasps, whether it's ice shifting off a glacier, what's fascinating to me is, you know, within hours, journalists have found some scientists who's devoted the past 40 years of their life to studying that exact thing, no matter how obscure it ends up being. And nobody cares about this period of this type of, nobody cares about coronaviruses until we've got a coronavirus outbreak. Nobody cares about killer wasps until they're suddenly rampaging across the Southwest. And so I love that aspect of supporting basic research, that we're creating this stable of folks. I mean, I don't mean to imply that they're only useful because eventually they'll be useful someday. I mean, there's this inherent, like, let's understand the world better. It will probably be useful in some fashion in the future. It may not be immediately useful, but it's cool to figure out how things are working. So yeah, I'm definitely even though I would say my lab is pretty fiercely focused on applications and on translation, on drug discovery these days, like really trying to be very practical about what's going to make a difference for humans in the next 10 years or something. We have a pretty short-term focus, I would say, in that sense, but I really appreciate the fact that there's basic researchers across the board studying all kinds of things. I think it's really important for society as a whole.
So what do you find the most fun aspect of your job then?
Most fun aspect. I mean, I guess I have to be a nerd in the sense that just seeing beautiful data and the social experience of sharing a view on some beautiful data for the first time is probably one of the highlights.
So there you go. There's
some lovely data. It's usually in the form of scatter plots and p-values these days, but
we do that as well.
Okay, yeah, there we go. So this shows the cluster of genes that you overexpress these genes and the cells look a certain way. And if you group cells based on the quantitative way that they look, this is what you get, a dendrogram that I mean, this paper was so exciting to me because this is the Robon eLife paper that was about overexpressing genes and using cell painting to cluster them. Basically, these clusters that came out of this experiment were recapitulating something, a tree of all these different pathways that have taken us biologists decades to figure out, who binds to whom, who's nearby whom in the pathway, who's upstream and downstream, which ones are negative and positive regulators of pathway, and just looking quantitatively at how the cells look when there's a bit too much of that gene present, protein product, they naturally will fall into these clusters. I just thought that was really a beautiful, that was definitely a beautiful moment when we saw this kind of data coming out.
I think it's one of the best virtual backgrounds I've had to date, because it kind of just makes it look like I've got a sort of crown or something.
Yeah.
It's like I
Yeah.
don't know. It's quite a different thing. I feel like a peacock.
Science is just radiant.
Feathers coming out the back or something. Which is certainly different. And that was interesting though, because again, you're a microscopist or image analysis, computer scientist, and we're looking at genes and how technologies are merging together. And that's a big challenge for you now, is to integrate, that spatial transcriptomics and the imaging, and putting the omics together with the imaging.
Yeah. So interdisciplinarity is the name of the game. It's hard to be an interdisciplinary scientist. I think, in general, if somebody's asking for career advice, the easier path is to pick a thing and be very good at it. When you're interdisciplinary, it means you just don't have time to be the absolute expert at, the 6 different things or 2 different things that you're trying to combine. And that can be a real challenge. And I think imposter syndrome is already rampant, but then you add on top of it the fact that you're trying to be an expert in two fields at the same time. It can be even more so. But I think the payoff is concomitantly better when you're trying to merge a couple of different worlds, because it's a space that others tend not to be as much in.
And I can't believe you used that phrase, because remember, just before we started, I was going to ask you a question which I haven't asked anyone else. I know a few brought it up, and that was imposter syndrome, and you just brought it up. So how confident are you? How often do you get imposter syndrome? Where are you most nervous?
So I'm glad you asked this, because it's such a fascinating thing to me that I feel I don't experience imposter syndrome in very many contexts anymore. I don't know if it's being just like in the Harvard MIT environment. It's just everyone is just so full of themselves and sort of you just sort of absorb it by osmosis. I don't know, but I really want to be clear that this was not the case at the beginning of my career. So I think I'm a good trajectory to follow if you're sort of down here at the moment in confidence level in your career. Maybe this story will help some of the listeners. So at the beginning, I think like almost every other kind of high achieving person in science, you have this one, well, like I look good on paper, but am I really all that? Do I have what it takes to survive in this career? Am I smarter than everybody around me? I had all those kinds of anxieties that I understand are really common for everyone. So what to say about, I could give an hour-long lecture on my progression through this trajectory to where I am now, which is not worrying about it. And I think a few things helped. Number one, I had a folder of all of my different accomplishments and certificates and awards and things like this earlier in my career stage, when that was more of a thing. And just flipping through that every once in a while remind me of something my best friend in grad school said, which is don't compare other people's outsides to your insides, right? Like you also look awesome on paper. You also have collected these awards. You also look fantastically amazing from the outside. And the only reason you think you aren't is because you kind of know how you feel on the inside. But that's a comparison you don't need to make. So that's one thing that was helpful. I think as well for me over the course of my career, it's been really valuable to me to have other sources of identity and value in my life. And so perhaps the most obvious one you could imagine is becoming a mom was one such dramatic experience that made me realize that just kind of cemented, does it matter if I'm the best scientist in the room or the smartest person or what? Like, does any of that really fundamentally matter, or are there other aspects of my life that give me meaning and value and so on? And I think for me as well, since the beginning, I became a Christian in high school, and underlying, I think my whole career has been this general sense that I don't have to perceive my value as other people see me, or how I'm assessed, or what my H index is, or any of these external, like it's part of the culture and the faith to not pay attention to these external sources of value, but instead to understand that each person is intrinsically valued and has purpose and meaning in the world, whether it's recognized by anyone else outside or not. Me living my purpose in life is really just between myself and my God.
This is cool. And So you felt that imposter syndrome. You've had a very successful career, but what has been the most challenging time in the career?
Most challenging career stage. No, it's all so challenging. It's all so difficult. I guess for me, it probably was just driven by my personal circumstances at the time. But I think starting up a lab was I think the hardest period, because I was also doing it while I was raising children. And so it was just like everything needed, you're building, in both cases, you're sort of building a solid foundation. Like, things are going to get easier in the future if you can just, if you can just get your first grant, and if you can just get your first awesome postdocs joining the lab, if you can just do everything yesterday, then things are going to be easier going forward. And the same thing with children. If I can just get them to sleep through the night, then, it'll be, so I just need to put in this energy now, and then things will get easier in the future. And so I do feel like now I'm living in that future where things have gotten easier, but that was a pretty crunch time for trying to build this nice foundation across a lot of fronts in my life at one time.
How did you cope with that stress? How do you partition it? How did you actually address the stress and move forward?
Yeah, I think it may be the same answer as the imposter syndrome. I think having dear friends who understand my situation, and then having this like source of value, knowing that I'm going to give it a try. If it doesn't succeed, if I don't get any grants, then I will find another job. And I want to be a scientist, and I love it's a pretty deep part of my identity. But I can be a scientist in other environments. I can not be a scientist at all. I can go into business and, feed my family if that's what I need to do. And so kind of trying to detach, it's hard. Like, I don't mean to make it sound like, who cares? I'll just find something else to do. But telling myself that, at least, kind of lowered the stress levels. If I don't get this grant, it doesn't mean, I'm a terrible person. It doesn't mean I'm a terrible scientist. It doesn't mean I'm unemployable. But it's just sort of coaching myself internally, I think, has been helpful.
So from fun to difficulty, what's the next big challenge? Where are you heading? What's got to be addressed? What's the big problem to solve? What do you really want to do? That's a lot of questions.
Yeah. So I think I do have it's a pretty broad and vague answer, but it's a pretty clear mission from this point forward. And that is to really push things more translationally and into the clinic. So as I said, roughly half the group now is working on the data science of images and the informatics of images. And as we've become increasingly focused on machine learning, it's clear to me that there are a lot of low-hanging fruit where the methods of machine learning applied to images. I mean, as microscopists and cell biologists, we love to look at images. We understand there's a lot of information there, but I think the information content is just so much more powerful than we really can absorb with our little brains. And so I'm so excited about
less of the little.
But I think really just being able to extract this information and set machine learning algorithms loose to be able to predict, so that we don't even, certain experiments we don't even have to do anymore. We can just say, hey, machine learning algorithm, look at all these images that I've treated with 100,000 different drugs and 50,000 different genetic perturbations, knocking genes up, knocking genes down. Take a look at all of that and tell me all the pathways that exist in the cell and how they interact with each other in this cell type, in that cell type, and so on. There's just a lot to be had there, and I know there's skepticism about machine learning in the biology world, and there's certainly some things that it can't do, some things it can do very well, some things it can't do, but I know there's a lot of low-hanging fruit, and it seems like we're just coming to the orchard and starting to reach up. So I think the next 10 years are really going to be about pushing things more in that direction of discovering medicines that are useful for particular disorders.
I would totally agree, actually. I actually think we can't do it without machine learning. I think it's the only way forward. But the question is, how long until it becomes commonplace to be using machine learning to understand more of our images?
Yeah, that's a great question. It's it's something you need data at a certain scale to be able to if you want to know if a particular gene is unusual, you have to test a lot of genes. Or if you want to know if a particular compound is remarkable in a certain way, it helps to have the baseline of what everything else looks like. And so it's not to say that it'll be everywhere in the sense that every lab will, all biologists need to be machine learning experts. But I mean, we've already seen quite a transition as to how computationally savvy most young biologists are these days. They certainly can enter a gene and blast and find a match and so on. Like there's certain tools that are becoming more and more commonplace. It's hard to find a biologist who hasn't used Image-J or CellProfiler to quantify their images if they take microscopy images. So I think it's certain tools are going to become really commonplace and really user-friendly. We won't really even think about them as machine learning tools so much. And others are going to be a little bit more restricted to those who have access to huge data sets and really the latest AI techniques. But yeah, definitely a wave is coming.
It'd be interesting to see how that is adopted. And we miss, I think the other important thing to mention is there's so much we miss in our images. And when we ask a question, or we are asking questions of our data, and we only look for what we want to see. And actually, I think machine learning will start popping out populations, trends that we are just blind to.
Yeah.
Right there.
And like you said, it's low hanging fruit. It's right there. It's right there for us to grab. So just looking at images in this way, I think there will be a lot that we can capture from it, even without really fancy techniques.
So what was the first microscope you ever used? Can you remember the first time you looked down a good microscope, a proper microscope?
I couldn't even tell you the brand of it. I guess it was probably a Zeiss or an Olympus. I don't know, but it was, a kind of, I can tell you what mattered about it, which is it was in a big dark room on a big table and had all kinds of contraptions hanging off of it and different pieces. And my job was to basically automate it, take up my microscope that was not intended to be automated in a real way. And I set it up to automatically capture images. And within the first week, the filter completely was like burnt out and, like starting to smoke. And so, I thought that, well, that's kind of unfortunate and odd that I just set up my microscope and the filter's burnt out, or the shutter had died on it. And I asked the manufacturer, like, can you send us another one? Because this one broke. And they sent another one. And then within a week, it was burnt out again. I called them back and they're like, what are you doing with that shutter? And the answer was, I'm running it literally 24 hours a day, 7 days a week. And they're like, yeah, it's not really designed for that kind of intensity. So yeah, that's what I remember about the first, big scope that I worked on. Yeah. Pushing it to the limits, let's say.
Yeah, I seem to remember mine having to pour liquid nitrogen in it to chill on the CCD camera, but that was some years ago now.
Yeah.
So some quick things. Thinking about your first microscope, what about your favorite items in life? So I'm going to ask, what is your favorite drink?
It's going to be water.
Does that turn to wine on an evening, or is it just water? And that's it.
Mostly water. I'm not. I really don't like wine. I somehow have managed to get through like college and grad school and postdoc and children, and have never gotten addicted to coffee. So I'll drink decaf, but just because it's warm and nice. But yeah, I can't say that I have like a serious favorite.
Yeah, it is decaf, by the way. So
what's that?
It is decaf coffee that I'm drinking.
Decaf coffee that you've got there. Yep.
Yeah. I do take caffeine, but only a couple of times a week, because then you really get the kick. So yeah,
it's actually worth it. Yeah,
it's actually worth it at that point. But otherwise, decaf, because you're right, I like the taste. Just, but I don't need the caffeine. I'd be nuts on caffeine. I'll be wired all the time. What about your favorite food? What's your favorite food?
Probably pad thai. So I grew up on a farm in Indiana, in the most culinarily like drab part of the country, I'm sorry to say, upbringing had no exposure to anything interesting. And so when I went off to grad school, and then of course coming to Boston as well, it's just been so exciting to be exposed to all kinds of different cuisines. So, but I know pad thai is not particularly the most exotic thing out there, but for me, it was quite a revelation.
And what about to cook? Do you cook, or do you prefer to be cooked?
Mostly baking, I would say mostly baking. It's got to be in the thousands of batches of cookies at this point in time across the course of my life, because I started when I was, 7 or so. So yeah, baking is definitely the big.
So what's your signature dish, or what's your signature cake or bake? Well, what is your?
So it's chocolate chip cookies. Yeah, it's pretty wholesome and American, but yeah, chocolate chip cookies for sure.
I have to come and visit if that's the case.
For sure.
I'm on board with that. So we talked about your favorite books. What about TV? Do you ever get to watch any TV?
Yeah. So I mean, there were definitely there were many years of my life where I didn't have a TV, and I was very, very proud of that fact. Now that I'm an exhausted parent, I do enjoy watching a bit of TV from time to time. But nothing nothing worth even commenting on. What do I watch? Like nothing. It's mostly just brainless kind of trash TV.
Yeah, but that sounds more interesting. Go on. What's your vice? What's your dirty secret with watching TV? What is it that you're most embarrassed about watching?
Oh, every once in a while I'll get sucked into one of these like relationship shows where it's, like however many, not The Bachelor, but those kinds of shows. But I will say, like, as a middle-aged person, it's much more fascinating these days watching people choose houses and then renovate houses. So I just remember my parents watching This Old House when I was a youngster, and here I am in that stage of life where I find it absolutely fascinating to see what happens when they knock down a wall and then they see what horrible electrical or plumbing has happened underneath.
You've just called yourself middle-aged. Do you realize that?
It's true. It is absolutely true. I'm 44. I don't know that I can expect to live much past 88. So yeah, there I am.
Do you not think of yourself as one of the young generation in science still?
Yeah, it's
early career scientist.
Science is such a funny thing because you're kind of raised on this, like, you have potential, and like at some point, you're actually middle-aged, or at some point, presumably, you become senior, and yet you still always have this mindset of like, I'm up and coming, I'm gonna be something someday. So it is a funny career in that sense. But I will say you do feel middle-aged when you notice that all the undergraduate students on campus, every year they get younger and younger and younger and younger, and you stay the same age, but somehow they are getting younger, and at some point the cognitive dissonance is too strong, and you realize, yeah, I'm actually a middle-aged person.
I think you also realize that I no longer wear fashionable clothes compared to what the youngsters are wearing. At that point, you realize.
But not because I don't know, it's because I don't care. And that's a really big distinction that I didn't have when I was young. I thought the old people just like didn't realize it, or didn't, couldn't figure out how to be cool. But I hadn't realized that they actually have other things in their lives they care about more. So that's kind of comforting.
I know the clothes I am comfortable wearing. It's almost like a brand, if you like. But so what is your favorite item of clothing?
Well, so now that we've been in pandemic for a year, it's clearly yoga pants. So that's for sure top front runner for me.
And what about music?
Music. All my music is kind of stuck from 15 to 20 years ago, when I used to actually sort of pay attention and to go to concerts and this sort of thing. So yeah, I would say it's just a collection of music from back then.
Okay, do you know, I've asked you what your favorites are. What's your least favorite food?
Liver was not great. I was told that it was a good thing to eat when I was pregnant, and I gave it a shot, but if my children are missing IQ points, it's because I couldn't stomach it. I
have to challenge that. Kidney?
I have not, can't say I've tried.
Surely that is worse than liver. I can get liver, but kidneys, that was just, as a child, that was the worst thing that I could, that was definitely not.
Growing up on a farm, we had our own cattle. So yeah, we were exposed to the whole gamut, but I can't remember. I think we always gave all the weird bits to my grandmother. So I don't recall ever having any of the sort of funkier parts that a person might want to eat.
So you just gave your grandmother all the awful food. I guess it's the same thing, isn't it?
It's the same thing, yeah.
So I guess if you were, I've got to be careful what I say now, because I'll offend some of the Scots here and their haggis with all the offal that goes into that. I know we've got Andy recording this for us that will be chuckling away and waiting to get his revenge on me after that comment about haggis and just being awful. I am joking, Andy. I actually really quite like haggis. We're actually coming up to the hour, which I cannot believe. It's been far too fast through that. As a last thought, for where would you like to see your career end? How would you like to see? Do you see yourself becoming emeritus and just keep going, or would you like to go out on a swan song and then go back and just sit back?
Interesting. It's hard. It definitely is hard for me to. I mean, I like to think that I'm not a workaholic. In the sense that as I said, I try to constrain my work hours, but I think I certainly have the elements of like, I would kind of like to keep doing this forever, because it's really an entertaining way to spend one's time. And so it's hard to say. It certainly would be nice to stay like deeply connected to science in some way, like off into the end years. But I like a lot of people, it's kind of fun to imagine just sort of winning the lottery so that you can self-fund your own lab and just sort of just focus on that for the rest of the time. So for me, I wonder if retirement might be a bit like that. Just latch onto somebody's lab where they can fuss about keeping everyone employed, and I can just maybe participate in some kind of loose way.
That's cool. And well, I will see you well before then, because you're not that middle-aged.
not quite yet. I'm not dead yet.
And thank you so much for joining us today. That's been absolutely brilliant. Thank you very much.
Really nice to talk to you, Peter. Take care.
Oh, just keep rolling for a minute. So they can edit in. So this is where you were brought up then.
Yeah, so my parents had around 100 acres of farmland in Indiana, and probably 6 or 8 different barns and outbuildings and so on. So that's one of them there, and it's just beautiful country. It's really a special thing to be able to grow up in the wild, so to speak. And that's another view of one of the pastures that's actually mostly wetland. So that's cattails you see in the foreground, but eventually turns into a bit of pasture.
Do you miss it?
I do. I mean, it's I enjoy not living on a farm on account of all the chores and work that need to be done, but it is delightful to visit for sure. And my kids absolutely love being there and seeing all the newborn kittens and interesting animals that are there. So it's a wonderful to have it be part of my life without having to actually handle it day to day. But that's where my work ethic comes from. It's no secret that to succeed in science is to some degree a function of just plowing through things that you don't necessarily want to do, just to kind of get things done. So that's where it's come from for me. So
genuinely farmer to farmer in that respect. You kind of got
that's right.
From agricultural farmer to pharmaceutical farmer.
That's right.
And thank you again.
Sure thing.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.